Hello and welcome. This recording is going to be discussing some of the anatomy that we find in the nephron. So the nephron is a microscopic structure. We definitely would not be able to see this on the kidneys of the cadaver, or if we were just looking at a kidney from the sheep that we usually do a dissection of now. But it is an incredibly important structure for the actual functions that the kidneys perform. So I want to walk you through the nephron at various levels. So at this sort of zoomed out level here, at this zoomed in level, and then what you would see if you were looking through a microscope. And like the liver pancreas video, I've got my little key over here that we will use to kind of indicate these various parts of the nephron. So what the kidneys do, what their job is to filter your blood. So you're going to have arteries that bring blood to the kidneys. The blood in those arteries will be filtered by all of these white structures that you see here. And then the new, cleaned, filtered blood would be returned to the body via some veins. So we're going to be discussing the white structures here, the renal structures that do the filtering of that blood. So because the filtering of the blood is going to be happening, and because that's a cardiovascular type thing, we are going to have parts that are cardiovascular and parts that are renal. So the cardiovascular parts would be things that we already discussed, blood vessels, so arteries, arterioles, capillaries, things like that, veins. The renal systems will be kind of our new systems that we haven't talked about. So the first thing to find is a structure called the glomerulus. And the glomerulus is a capillary. So it's a capillary bed that does the filtering of the blood. And so here we see an artery, and this artery leads up and then leads into this kind of Christmas tree looking structure. This is the glomerulus. So the glomerulus is our fancy term for a capillary bed that filters the blood in the kidney. Surrounding the glomerulus is this structure right here. That would be this white structure right here. This is the glomerular capsule. So this is a structure that's going to be collecting the filtered plasma out of the blood here and taking it through a series of tubes that will eventually lead to the bladder and out the body as urine. So this, indicated by this number, by this letter A here, is the glomerular capsule. So the glomerular capsule collects that filtrate and then goes into this twisted tube here. This twisted tube is the proximal convoluted tubule. So it is the tube that's closest to the glomerular capsule, and convoluted means twisted. So it's the close twisted tube. This twisted tube then leads into this structure. This is the nephron loop. So all of this is the nephron loop. You can see it's a loop structure. And the loop structure will lead into this structure right here, which is another twisted tube, only this one is far away from the glomerular capsule. So this is the DCT, or the distal convoluted tubule. Our distal convoluted tubule then leads into this big structure right here. So many DCTs lead into this structure from all over the place. And this structure is the CD, or the collecting duct. So any blood that got filtered out and flowed through the PCT came through the loop, flowed through the DCT and eventually gets to the collecting duct will most likely be leaving the body as urine. Should probably be yellow, but we work with what we got. So the glomerulus is cardiovascular. It's the red Christmas tree looking ornament type thing in there, the capillary system. The white around it is the glomerular capsule. This twisted tube is the PCT. This big loop is the nephron loop. This twisted tube is the DCT, and this long big tube with many DCTs connected is our collecting duct. Now in the next picture, what we're going to be doing is we're going to be zooming in on this right here. So this interplay of the glomerulus, the glomerular capsule, and the DCT right there, as well as this artery bringing blood into this structure. So this is just this guy, only much more zoomed in. What we can begin to see here are some more specifics. So up here we saw that an artery right here, this guy, led into the glomerulus. And if you look elsewhere on this diagram, you can see there is an artery or an arteriole coming out of the capillary bed. So here we've got the one going in, and then one coming out. We've got the one going in, and then one coming out. So there are two arterioles that lead in to the glomerulus and then out of the glomerulus. And these two arterioles are the afferent arteriole and the efferent arteriole. So this is our afferent arteriole. This is the one bringing blood to this capillary bed where it will get filtered. After the blood has been filtered, most of it is going to leave by being picked up again and shot out of this arteriole. This is our efferent arteriole. This is the afferent one. This is the efferent one. This is the afferent arteriole. This is the efferent arteriole. Your afferent will always have this structure here, this big clumping of cells, and that's one way that you can tell if you're looking at the afferent or the efferent. So the afferent arteriole brings blood into the glomerulus. That would be this red structure. And this red structure continues underneath these white cells here. So this entire thing is the glomerulus. And we can see that we've got the glomerular capsule right here, all of these cells all the way around. So all of these cells are the glomerular capsule. And we've now got a kind of new structure that we can see as we have zoomed in so close. We see that between the glomerulus, the capillary, and this kind of shell that covers it, the glomerular capsule, we've got this open space, and this is the capsular space. So this is where the blood filtrate that got pushed out of the glomerulus is collecting inside of this open area. So anything that gets pushed out here collects in this open area, and it will begin its journey. We said that it began its journey once it gets filtered in the PCT. So this right here must be our PCT. So there's our proximal convoluted tubule, taking whatever got filtered out in its journey through the tubes, eventually to the collecting duct. Now we have one additional thing to add here. Notice on this picture, we've got the loop, and then we've got the beginning of the DCT. Right at the beginning of the DCT, it begins right near the entrance of the afferent and the exit of the efferent arteriole. As we look, here's the entrance of the afferent, and there's the exit of the efferent. So this must be the beginning of our DCT. And in particular, there are a group of cells, these right here, called the macula densa cells. So these guys are just this strip of cells on the DCT here. And they're very important in sensing solute concentration and blood pressure and adjusting not only the rate that filtrate is made by the glomerulus, but also adjusting your blood pressure through a number of different mechanisms that we will not talk about here, but if you come take physiology with me, we'll talk about them then. So basically what these cells are, these macula densa cells, are blood pressure and solute sensing cells. And so they're going to be kind of sensing blood pressure in a roundabout way. So these are our model versions of the kidney. So this is the cortex up here. This is the medulla. These are all the things you'd find in there. This is a blown up version of the glomerular capsule and the renal corpuscle and all that kind of stuff. What would this actually look like if you were looking through a microscope? So what it actually look like is something like this here. So we are going to find just parts of what we found up here in this picture here. So the first thing that we can find, hopefully we can see all of this junk here. This would be our glomerulus. So this red structure is all of this twisted mess that you see inside of here. So this is the glomerulus. That would make this area right here, this open area, that would be our glomerular, that would be our capsular space. So this is the space that the filtrate is being made, and then it's being captured by the structure that surrounds the glomerulus, which would be this structure right here. So that would be our glomerular capsule. So we've got the glomerulus, we've got the glomerular capsule, we got the capsular space. Now, three other things that we can find are macula densa cells, we can find the PCT, and we can find the DCT. So, first off, macula densa cells. Where were they here? Well, they were at this kind of end of the glomerular capsule where the arteries are coming in. You can't really see those arteries very well, but you can see a very nice lumen with some simple cuboidal cells very near an opening of the glomerular capsule. And that's going to be right here. So these guys right here will be our macula densa cells. So there's the macula densa cells right next to the big glomerulus. Now all throughout here, these various tubes are going to be parts the PCT and parts the DCT. What really happens when the tube leaves the glomerular capsule, so let's say the glomerular capsule is in green here. When the PCT leaves, which we'll draw in orange, it comes out and does this, so kind of a big mess. Then it goes down through the loop and comes up and becomes the DCT, which we'll draw in red. Now the DCT isn't in another place. The DCT is a big mess all around in here as well. So basically, if you take a histology slide of this, what you have is PCT and DCT right next to each other. And so all of these lumens that you see all over the place in here, some of them are DCT and some of them are PCT. So how do we figure out the difference? So PCT, these guys are going to be more red and fuzzy, especially in the lumen, and that's because they have microvilli. So our PCT, here is a PCT, very red. You can see some fuzziness on the inside there. Here is another PCT right there. Here is another PCT. So these all would be PCTs because of the deep red of the cells and the fuzziness in the lumen. Our DCT, which we do in red here, are going to be lighter and have a clear lumen. So here would be a DCT, here would be a DCT. So you can see that lumen is much clearer than it is over here. So right at the edge, there's not that fuzziness going on in there. So this video then goes over the histology and models that I'd like you to know for the urinary system.